Good morning, students. Today we start the new chapter, that is plant growth and development. What is growth? All cells of a plant develops from the zygote. A zygote produces a number of cells which organize into tissues and organs. Development is the sum of two processes, growth and differentiation. During the process of development, a complex process takes place. Growth may be defined as an irreversible permanent increase in size, volume or mass of cell or organ or whole organism. Growth is one of the fundamental characteristics of all living beings. It is accompanied by metabolic processes, that is anabolic and catabolic process, that occur at the expense of energy. Characteristics of growth. The main characteristics of growth are cellular growth, cell division, cell expansion, and cellular differentiation. Growth rates. Growth rate can be defined as increased growth per unit time. The rate of growth can be expressed mathematically. Types of growth rate. There are two types of growth rate, arithmetic growth and geometric growth. Arithmetic growth. After mitotic cell division, only one daughter cell continues to divide while others differentiate or mature. Example, root elongation at constant rate. Mathematically, it is expressed as LT is equal to L0 plus RT. Geometric growth. Initial growth is slow, that is a lag phase, followed by a rapid increase in growth, that is the exponential phase, and followed by a phase where growth slows down, that is a stationary phase. That is a sigmoid S curve type of graph in the case of this geometric growth. If one plots the parameter of growth against time, it would be typical sigmoid S curve. The exponential growth can be expressed as W1 is equal to W0 ERT. W1, that is a final size, weight, height, number, etc. Differentiation, dedifferentiation and redifferentiation. Cellular differentiation is the process by which a less specialized cell becomes a more specialized cell type. Cell derived from meristems and cambium differentiate and mature to perform a specific function. Differentiation. Cells undergo structural changes during differentiation. Changes take place both in their cell walls and protoplasm. Example, cells lose their protoplasm during the formation of tertiary elements. Plants develop a strong elastic lignocellulosic secondary cell walls to carry water to long distance. Differentiation, dedifferentiation. Dedifferentiation is an important biological phenomena whereby cells regress from a specialized function to a simple state remains of stem cells. Redifferentiation. Redifferentiation, differentiated cells divide and forms the new cells. Cells produce and lose the power of division and may be the part of permanent tissue. Development. Development is a term that includes all changes that an organism goes through during its life cycle from germination to the seed to senescence. Plasticity and heterophyly. Plasticity, the ability of plant to follow different pathways and produce different structures in response to environment and phases of life. In cotton and coriander plants, the leaves of the juvenile plants are different in shape from those mature plants, that is heterophyly, presence of two types of leaf in same plant. Now see the plant growth regulator or the phytohormones. Plant growth regulator, that is phytohormones, are chemically substances that influence the growth and differentiation of the plant cells, tissues and organs. Plant growth regulators function as a chemical messenger for intercellular communication. They work together coordinating the growth and development of cells. They could be indole compounds, indole 3 acetic acid, adenine derivatives. There are 5 types of phytohormones: auxin, gibberellin, cytokinins, abscisic acid, and ethyl alcohol. Classification of plant growth regulators, that is divided into two: growth promoter and growth inhibitor. Growth promoter hormones are auxin, gibberellins, and cytokinins, and growth inhibitor hormones are abscisic acid and ethylene. Auxins, that is a growth promoter hormone, first of all discovered by Charles Darwin and Francis Darwin. F.W. went isolate auxins from the tips of coleopetals of oat seeds. Went developed Avena curvature test for bioassay of auxin. Synthetic auxin is the indole acetic acid, IAA. Physiological effects of auxins: promote apical dominance, initiate rooting in stem cutting, promote flowering in pineapples, prevent fruit and leaf drop at early stages, promote the abscission of older mature leaves and fruits, induce parthenocarpy in tomatoes. Now second is the gibberellin. E. Kurosawa, a Japanese scientist, identified gibberellins present in a fungal pathogen Gibberella fusicori. Gibberellins were isolated from infected rice seedling when treated with sterile filtrate of fungus. Physiological effects of gibberellins: elongation of intact stems, dwarf shoots, bolting of rosette plants, overcome the natural dormancy of buds, tubers, seeds, etc., and seed germination. Cytokinins. Skoog and Miller discovered cytokinins. Skoog and Miller crystallized the cytokinesis promoting active substance, named it kinetin, from corn kernels and coconut milk. Physiological effects of cytokinins: induce cell division, delay the senescence of leaves and other organs, promote lateral bud. Ethylene. Cosins confirm the release of volatile substance from ripened orange that hastened the ripening of stored unripened bananas. The volatile substance was identified as ethylene. Physiological effects of cytokinins: promote senescence and abscission, inhibits elongation of stems and roots, induce fruit ripening, breaks seed and bud dormancy, initiates germination in peanut seeds. Abscisic acid. In 1963, Frederick T. Endicott and his co-worker identified abscisic acid. ABA was isolated from several abscission accelerating substances from cotton plants. ABA is called stress hormone. Physiological effects of abscisic acid: acts as a general plant growth inhibitor, induces seed and bud dormancy, inhibits seed germination, stimulates the closure of stomata. Photoperiodism is a response of plants to the relative lengths of light and dark periods. Some plants require a periodic exposure to light to induce flowering. Duration of dark period is equally important for flowering. Long day plants Plants that require exposure to light for a periodic exceeding critical duration to induce flowering. Example, pea, barley, wheat. Saute plants, plants that require exposure to light for a period less than this critical period to induce flowering. Example, rice, jowar, cotton. And day neutral plant, plant where there is no correlation between the period photoperiod. Vernalization is the phenomena of induction of flowering in plants by exposure of low temperature. It prevents precious reproductive development late in the growing season and enables the plant to have sufficient time to reach maturity. Example, biennial plants. These are monocarpic plants that flower and then die in second season.